Blog Talk Radio.
Welcome to the AME Food Testing Show. Today's topic, detection of pathogens in food production fields, with Laura Strawn, Master of Science. Laura Strawn has received several awards and honors from professional associations such as the International Association for Food Protection, the Institute of Food Technologists, and the Food Science Department at UC Davis, also at Florida and Cornell universities. She's published 4 peer-reviewed articles, 1 book chapter, over 10 abstracts, and organized and chaired 3 symposia at the annual meetings of the International Association for Food Protection. In 2012, she was also awarded a National Institute of Food and Agriculture Predoctoral Fellowship grant to conduct her own research program that focuses on the safety of produce in the pre-harvest environment. Laura has also been a teaching assistant for multiple classes and received the Outstanding Teacher Assistant Award at Cornell in 2010. She will graduate this year with a Ph.D. in food science with specialities in microbiology and epidemiology. Now, welcome with me, Laura Strawn from Cornell University. Laura?
Hi, and thanks, Andy. That was really a kind introduction.
Well, would you like to add anything as far as an update to our audience of 37,000 food production, food quality, food safety, and food security managers?
I think you pretty much covered everything in terms of the highlights about me.
Well, Laura, welcome to the show. Let's begin with our first fundamental question. What is a foodborne pathogen?
A foodborne pathogen is basically any agent, such as a bacterium, virus, parasite, that can cause disease where the vector is a food medium.
Very good. What have been the traditional methods of detection of foodborne pathogens?
Pretty much the traditional methods have all been culture-based, where a sample is taken and enriched in a certain culture media, and that is the initial growth period. And then from there, traditionally they've used more selective enrichments and then just simple plating on selective and differential augers. Now, as we've entered into new technologies and everything changing so quickly, they've come up with more rapid methods. And anything that would be described as rapid would just be increasing the time that it would take you to detect these pathogens. Still, with the rapid methods, you do have a enrichment in culture media. It's just significantly shorter.
Very good. So that's how we find out whether or not there's a pathogen in that foodborne or food matrix. Let's find out a little bit more about your studies. What are the traditional methods to trace back pathogens to production fields?
Currently, right now, if you do have a foodborne outbreak, epidemiologists, government agencies, it's really a collection of people that work to determine the source. And if it is traced or they believe that it's to an implicated farm or production facility, they'll actually go out there, they'll collect samples, and then from there they'll test. And they'll either find that they have a negative result or positive, and from there they'll continue on with their investigation. Just because you find a positive result, that would be maybe highly conclusive that may be the source, but not always the case.
Well, Laura, let's talk about some of those methods. What do they use on a traditional method to find out if there's a pathogen in the production field?
If they're doing environmental tests, go ahead.
Do they collect water, soil, The actual plant producing the food? Go ahead.
So if you're going out to an implicated farm, the first thing that they'll do is pretty much pull out a map of the farm, sit down, and decide what are potential areas that may have contributed to where there's points in the system that could have broke down. For environmental samples, they'll collect agriculture, water, soil. They will collect produce, or actually like a sample if necessary. And in terms of from there, they'll go into maybe a packing house or further down the line.
So your study at Cornell University is a new method for the detection of pathogens in food production fields. Could you outline your study for our audience?
Sure. Basically, with our study, we wanted to use a combination of environmental testing and taking that one step further with using GIS-based technologies to really set up a framework for if we can detect these pathogens in the environment and be able to predict areas that maybe represent lower risk for your produce grower. And the way we set up our environmental testing is we went out and we collected water samples, soil samples. We used a drag swab, which is a great way to collect any kind of topsoil or potential pathogens maybe that are on the surface. We did not collect produce samples for our study. And that was for a various amount of reasons, because we wanted to really focus on the environmental aspect of potential contamination. We also did collect fecal samples that we possibly found within the pretext of the designated areas that we went out to sample. We sampled fields, so we tried to standardize the size of the fields to give weight to the study. And from there, we went out and we collected samples in 4 times a year. So we went out seasonally, fall, winter, spring, and summer. And that way we could kind of be able to answer if there was a particular time period in the year that you may be more or less likely to be at risk. And from there, after we collect the samples, we use traditional culture methods to detect and isolate any potential foodborne pathogens. We looked specifically at Listeria monocytogenes, Salmonella, and Shiga toxin-producing E. coli, so your S-Tex. And from there, we also, every single collection site, we took a GPS coordinate. And that way, we could obtain remotely sensed data. There are a huge data bank of public available information on soil properties, land use designations, and of course, with the date of the sample, you can pull up a huge pool of information on meteorological factors, so precipitation, temperature, whether it freezes, or etc.
Well, I have a couple of questions for you, Laura.
Okay.
Did you finish, by the way?
Yes.
Okay. What is GIS Technologies? You mentioned that originally in your start of your discussion about your method. Can you review that?
Yeah. So it would be GIS stands for Graphical Information Systems, and through GIS, if you have a GPS coordinate, there are public available software where you can basically take a GPS coordinate and using various programs, you can pull up you can basically input this remotely sensed data. So if you're looking at a particular environment and you want to know where in this landscape the soil pH is high or low, this will really be able to focus in your areas and actually be able to kind of produce maps that will show you areas where the soil pH is a certain cutoff for you, or that here are your land use, that you might have a pasture or type of area, and this is in relation to where your farm is. And all of this is done for the spatial characteristics like soil or landscape via satellites and many government-funded operations where they go out and survey. So that's kind of the combination of wrapping all of it up into kind of a box in a very concise way.
Well, Laura, that's fascinating information. Now, when you said pull up, do you mean find it on the Internet and then use it in your own operation without a charge? It's government accessible information?
Yes, it's all available.
Find that?
Excuse me?
I said, where would our listeners find that information?
Basically, if you want to look up any kind of meteorological data, so you're interested in finding out past temperatures, precipitation, you can actually access all of this information online through the National Oceanic and Atmospheric Administration. There's the National Climate Data Center and a local climatology database. And from there, it basically pulls up, for example, a map of the United States, and it has all the different weather stations, and you can focus in on your specific area that you're looking for, and they may have a airport weather station that they use or a local weather station, and from there you can click on that and actually input like the dates that you're looking for and pull up all of the information that way. And it'll actually send it to you to your email address, and it'll basically open in a large spreadsheet that you can open in Excel or any kind of open access type of Excel spreadsheet that you have. With soil data or landscape features, there you can use through, if you're looking for a land use, like a pasture, proximity to forests or impervious surfaces, which is just roadways and urban development, you actually can use the National Land Cover Database. And they obtain all of this information through different satellites. And from there, you can pull up any of the images that they've captured, and so you can really get an idea of an accurate representation of what you're looking at landscape-wise. For your soil properties in terms of pH or whatnot, those can be acquired from the U.S. Geological Survey. You can do it through Earth Explorer, which is a geographical database, and you can also obtain various, as I mentioned before, map layers where you can pull up different soil, water storage capacities, the pH, through the U.S. Department of Agriculture Soil Survey geographical database. And all of these are publicly available. All you have to do is log on and kind of know where to start hunting for the information. And all of this is completely open access. And from there, you can download almost any host of information that you're looking for.
Well, Laura, your information is actually a wealth of information for our listeners, particularly those that are involved in direct food production. Let's review quickly, what is your methodology for drag swab of the soil for looking for surface contamination? I find that to be a very fascinating protocol.
Yeah, so previous literature, they've come up with this drag swab, and it's really actually quite easy to make this particular drag swab sample collection type method. We used 2 pieces of gauze, so just sterile medical gauze that you can buy from a supplier. Once you have the gauze, you basically fold it in half and kind of tie it with a sterile string. This string you can get from, a Home Depot or a Lowe's, and from there you'd want to sterilize your string, and you just tie the gauze together and have about a 6-foot long string that you can pull it to the side of your body, so you don't pull it behind you or whatnot, where you're going to possibly contaminate your scene by whatever, if you're walking with your shoes. But yeah, it's extremely easy to make. It's, as I said, in previous literature, if you actually read my paper, we do cite the previous literature in there, and it's a pretty sound method in the community. It's a good technique to use, and as I said, it's very affordable. And in terms of what we're looking for, it worked.
So in this drag swab protocol, are you swabbing areas that you suspect you might have animal intrusion or some other way for E. coli, salmonella, listeria to contaminate your production area? Is that the intent?
Basically, that's the intent, to capture anything in the topsoil. When we took soil samples, we actually went more we did about a minimum of 6 inches under the surface of the soil. So you were using a sterile scoop to kind of dig into the soil and, really be able to capture what's underneath the soil and what's on top.
Excellent. Let's talk about the seasonality of your testing. I find that particularly compelling since the food production cycle is pre-harvest and harvest. And that's pretty much it as far as I'm concerned. But you are, in your study, we're looking at fall, winter, spring, and summer. Can you explain that seasonality of testing?
Yeah, and that was basically to capture, depending on where you are in the country, it's always a little different. For example, in Florida, when I was down in Florida, they do a lot of their harvesting almost later in the year, if you're just looking at a regular year from January to December. Somewhere in New York, where we actually have a very different winter than what they have in Florida, the harvest period is actually earlier. So we wanted to kind of really capture a wide range. We didn't want to look just at summer or just at fall. We wanted to kind of, yes, the study took place in New York, but we wanted to be able to kind of have different academics or industry be able to see this framework in place and feel like it could be applicable or they could draw on conclusions for their own geographic area. And we used basically an astronomical season type window that we looked at. And additionally, here in New York specifically, with the winter, you do get a lot of freeze-thaw. And freezing and thawing, you're getting sometimes when it thaws, you have an excess amount of water that we wanted to see really how that impacted the environmental ecology of these foodborne pathogens.
Fascinating. Let's talk about your use of GPS coordinates. How does that help in food production management identification of areas of concern for food pathogen intrusion? So with a GPS coordinate, it's basically, as I said, you can pull up an entire amount of remotely sensed data with this GPS coordinate. But from there, once you have these factors that you've identified for your specific farm or geographic area, from there, you can actually almost use a precision-type agriculture way to identify, okay, you're going to plant your block of commodity. Well, using these GPS coordinates, you can go out and know exactly where these potential risky areas are or where the low-risk areas are. And that's one of the things we really tried to focus on in the study is that we can identify low-risk areas that the risk is extremely low. And because
there obviously is a certain amount. We know that the environment, there are going to be pathogens in the environment. It's not a closed system. It's the environment. Bacteria, they live out there. But where they are is not uniform. You don't have a uniform risk everywhere in your farm. So we wanted to be able to say, hey, this is a specific area or set of locations that you have a really low, a lower prevalence of these pathogens, in this particular location. And using GPS is a way that you can focus it.
Excellent. And you've already alluded to it, what are the findings of your study that can be applied to food production management?
So one of the things we found, as I said, is that contamination or these pathogens in the environment, they're not uniform. You don't have the same risk everywhere on your farm or in your geographic location, which, for example, in the New York study was the central New York to western New York area. And so that was one of the first things that we wanted to establish. And once we established that, we figured, could we identify areas of low risk or potentially a little bit higher risk? And that was when we decided that we had collected all this remotely sensed data using a method in statistics, and they use it a lot in landscape ecology, these classification trees. They're a nice way to take all of this information and put it into a system where it'll take your detection of a positive sample or a negative sample in combination with all these remotely sensed data characteristics, your soil pH and whatnot, based on all the locations that you took these samples, and it'll, in a nice visually appealing tree, it'll set out rules. It tries to group positive samples in one cluster and then negative samples in another based on a factor. And so we'll break it up into pathogen, because we did find that there are various factors for each pathogen. There's not necessarily an umbrella type set of rules for what you're looking for. So with Listeria monocytogenes, for example, we found that one of the major set of rules was proximity to various land uses. We found that if you were within a certain distance of proximity to a waterway, so this could be a surface water source like a creek, it could be a pond, and what we found from there was that if you were within this certain distance, you almost had a 4 times more likely to find a pathogen-positive sample than a negative. And so what that's telling you is that if you're closer to water, you have a little bit of a higher risk than if you're further away from water. And one of the other things that we found in terms of these proximity to these land uses were proximity to urban development and roads, so the impervious surfaces, which we found that there was almost it was about a 12% prevalence if you were extremely close to an impervious surface versus only about 1% predicted prevalence if you were farther away. And that in itself maybe gives way to some of the distances that they don't plant crops right close to the roadways or right up against maybe somebody's house. So that, that kind of intuitively made a lot of sense to us. Another one was pastures. And one of the important designations to make is when you say the word pastures, everybody wants to think a dairy farm or a beef farm. And through this remotely sensed data, what they're defining as a pasture could be an animal operation. It could be just a large stretch of land that's used for these animals to graze in terms of a pasture area. It also could be designated as a hay environment where it's just a huge kind of strip of land that, again, is set up for the particular grazing of animals. And that's an important designation to make. So just because that you may be close to a dairy farm, we don't want to pick on any of those types of operations because that's not what the remotely sensed data is giving you. It could just be designating a pasture area. So we found that if you were within a certain meters distance from one of these pasture areas, which could just be where the animals graze or could be where they might have grazed 50 years ago because land uses are always changing in today's climate, we found that you had just a much greater risk of contamination in your pre-harvest environment in these environmental samples than if you were farther away.
Well, Laura,
so one of these.
Oh, go ahead. I'm sorry.
I was going to say, and if we wanted to look at the temperature side, we also saw that if you had cooler temperatures, you were more likely to find a Listeria monocytogenes positive sample than if you had warmer temperatures. So if we wanted to look at the temperature aspect, that was one of the particular results that came up.
Well, can we just take a step back to the animals? I found it particularly compelling when I compare your data set with the findings in Monterey County in 2006 with the spinach E. coli outbreak because of the feral pigs or the wild pigs that were grazing on the spinach and yet tromping through a cow pasture where there were cow patties and obviously transmitting them with their hooves from the cow pasture onto the production areas. So your data set there is particularly compelling. Let's talk about the cooler temperatures. I find that particularly interesting. You're talking about normal temperatures throughout the year being cooler. Is that correct?
Yes. Basically, whatever so in New York, what we found with the temperature is if you look at previous years, they have information on the average temperature for that day or for that period. And what we found is that if you were below the average temperature from previous years, that was when you had a higher predicted prevalence of particularly finding a pathogen-positive sample. And that's one of the important things I want to note, too, is these are potential pathogen-positive samples. And again, this isn't samples on the produce. This is just environmental, that if you took a soil sample, you may have a greater likelihood of finding a positive sample. And that's important to note because there still is a lot of research going on out there where just because you have an environmental sample that is found positive, we're not exactly sure yet what exactly that means in terms of how likely your produce is to be contaminated. And that's something that there are many people out there researching that and trying to give scientific-based findings on that. And that is something to note is that we're not clear on what that exact, point A to point B is.
So that correlation between environmental sample result and production contamination, that equation hasn't been really finalized yet, is what you're saying.
Yeah, it really hasn't. I think that there's a lot of information going on out there, and probably the norm would be, we really don't want to find any environmental particular sample pathogen positive. We don't want to have that in our pre-harvest production environment. But just because you find a sample one day, you could go back out the next day and it could be negative. So we're not able to distinctly be able to have a causative link between an environmental sample being positive and what that risk is for your produce to be contaminated. And that's an important designation to look at. We want to be as preventative as possible, but in terms of a causative link, we don't have one, and we don't have information, quantitative-based information, to really make a designation or a clear-cut A to B. There isn't a clear answer for that.
Well, Laura, let me congratulate you first on the intensity of your study and the inclusion of the publicly accessible data. That's called transparency in academic parlance. I would ask you a question, and that is, how can a food production manager apply your study? Or is it something that crop scientists would apply in their individual studies of a field for food production?
First of all, we definitely want to validate our GIS modeling framework, and that actually is something that here at Cornell University, and before I graduate, I definitely want to set up a validation study. I'm in the process of trying to formulate a really good experimental design that's robust to validate some of the rules and factors that we found in this previous study that was published in the Applied Environmental Microbiology Journal. And from there, once we have a validation study to show that, yes, we can predict locations of higher prevalence versus a lower prevalence of pathogens in these environmental samples that we take. From there, I think that the produce grower probably will be the most beneficial recipient of this information, because from there, what we want to be able to really get at is that if we can identify these areas that are of extreme low risk, from there possibly you could plant a commodity that is consumed raw, that we are, is more of a high-risk crop, whether that be leafy greens or tomatoes. A grower then can say, okay, maybe in this area, because we have sound scientific data, again, that would need to be proved with validation and with more data, other places in the country using our study as a framework to really give, as I said, a robust conclusion to this data that this does work. Once you could identify these areas where it's very low risk, then as a grower you could choose to, if possible, plant more of these high-risk commodities in these low-risk areas.
That's an excellent conclusion to today's discussion, that your study is a preliminary study. There's still further research that needs to be accomplished before we can actually focus in on some definitive findings. But I would really congratulate you today for your participation on today's discussion and allow you to interject any final conclusion comments.
Basically, if I wanted anyone to take anything away from this, is that the produce, pre-harvest safety it's extremely challenging. It's not something that's easy. Growers, they have a tough job. They have to account for all of these different land combinations. And I think in general, even going out, to, as I said, I haven't been all over the country, but here in New York, they do an excellent job. They really care about food safety. They're really interested in the science. And from there, with that particular combination of growers caring about safe food, doing an excellent job, with academics trying to answer some of these questions, everybody is concerned about it and is really diligent. And I think that is an amazing combination, because that's where results are going to come. It's from teamwork and working together and sharing information and being open to ideas. And from there, I think, it's only going to get better. And we already have a safe food supply, so if you can even make it safer, then that's something to really be proud of.
Well, Laura, I'd like to congratulate you on your future Ph.D. awarding, which will come later this year, and also thank you for your participation today on the AME Food Testing Show and your contributions to the food production area, and look forward to your future contributions to our industry particularly for those involved in food production, food quality, food safety, and food security. And again, thank you for your participation today.
Well, thank you for having me. I hope to be on again, maybe when we have some validation results.
Yes, you were invited to come back and share with us your validation results and perhaps some specific applications and some lessons that we can draw and apply toward our industry to make a safer food production environment for all. Again, thank you, Laura, and wish you a great day.
Thank you, and have a nice day as well.
Yeah, bye.
Bye.